All right, so in the next talk, we're going to continue on with the fungal topic with Rytas Vilgalys. And for those of us who work on microbial communities, we've been somewhat forced to steer clear of the eukaryotes for a long time, and a real goal has been to incorporate the eukaryotes into our work. And so this is a particularly timely talk, talking about actually starting to do environmental studies of fungi associated with trees and forests. So Rytas Vilgalys from Duke University will be talking about understanding the forest microbiome from a fungal perspective.
Thank you. Thank you. And I'd like to also thank the organizers for inviting me to give a talk. I've had the good fortune to interact with people from JGI for a number of years now, and also collaborating on a couple of projects that are directly related to DOE and JGI, as well as some NSF-funded research that I'm going to talk about today. So today I'm going to tell you a little bit about two of those projects, one on the microbiome of Populus and the fungi that are involved, and a second project that is dealing with the microbiome of pines and why they're interesting. So the first project is one that's led by a team from Oak Ridge National Labs with Chris Schott, Jerry Tuscan, and others. And on the right, you see Greg Benito and Khalid Hamid, who are Duke scientists working on the fungal side of these interactions. And this project is a rather tour de force, examining the microbiome, bacteria and fungi associated with the rhizosphere of plants, and then trying to examine the interactions between these organisms within the rhizosphere using genomic and metabolomic tools. So let me just tell you something real quickly about Populus. It's obviously a very important flagship species for the Department of Energy, and the first tree species to have its genome completely sequenced. Populus is mycologically interesting because it has a number of diverse interactions with many different kinds of fungi, including Ectomycorrhizal and Arbuscular mycorrhizae, as well as a large number of endophytic fungi. Endophytes are fungi that can grow within plants, but are often asymptomatic. So they're still kind of a black box in terms of what they're actually doing there. So our interest has been to characterize these different fungal guilds that are associated with Populus. And these can range from saprobic fungi, which are primarily decomposers, to the two major mycorrhizal guilds, which are symbiotic associates involved in nutrient exchange and uptake of plant photosynthesis. And as I mentioned, the endophytes, there's a diverse number of these fungi, but we still don't really know what their functions are. And then, of course, there's a huge array of pathogenic and parasitic fungi occurring on these plants, and those are better studied. So some of the general questions that we've used have been basically using field campaigns, surveying the rhizosphere, collecting root samples, and then making isolations of bacteria and fungi to bring these interactions into the laboratory. So I'm going to tell you something about the fungal side of these interactions today. If you have a chance, a number of posters at the meeting here are describing some of the ongoing work by Chris and others, starting with our studies on Populus deltoides, primarily, as you see, in the eastern part of the US, and comparing sort of the biogeographical and host variation across this range. And there's a number of papers here describing that work. More recently, the group has focused its attention on some other sites on the West Coast where Populus trichocarpa is the dominant tree species. In particular here, we're looking at a common garden study involving 1,000 cloned Populus genotypes planted into 4 different common gardens from California up into British Columbia. And again, looking at a number of different aspects involving lignin production and also the heritability of microbial communities. I won't be talking about any of that because my contribution from our lab has been to basically take these soils from these sites into the laboratory and use the plants to trap the fungi for us. This has certain advantages, particularly when we are trying to isolate the fungi. We can grow them in replicated containers and then examine those interactions in a repeatable fashion. So we typically grow our plants, and you can see here's Populus on the left, but we've also looked at some other host species, particularly pines and oaks, because we know that those are strong formers of Ectomycorrhizal on their roots. We harvest those roots, we wash them, and then we use next-gen sequencing to look at the fungal communities that are present. And we're just starting now to look at the metatranscriptomes of these root systems to identify how these fungi are functioning within the plants. And then because we've been isolating fungal cultures, we are also trying to develop systems for looking at Populus fungal interactions. And not just that, but also looking at fungal interactions with the bacteria from these same habitats, same niches. And again, I won't be talking too much about the bacterial interactions. So to summarize a number of studies, this is one of our initial experiments, was basically using the bioassay to ask how distinct is the fungal community on Populus with respect to other potential plant hosts. And as this ordination here shows, Populus is quite distinct from oak and pine, which are actually share a fair bit of their fungal community in common. And this is largely due to the fact that oaks and pines are highly ectomycorrhizal. And you can see that here too, because both Populus has a larger fraction of unique fungal taxa associated with its roots, in contrast to these other hosts. When we look at the fungi that are there, a couple of things stand out right away. Again, we're working on Populus deltoides or cottonwood, and what we've been finding consistently is that it's a very poor former of Ectomycorrhizal. It associates with Ectomycorrhizal Basidiomycetes and Ascomycetes, but by and large, the roots are often naked and associated with other fungi. And so I've just simply starred them here. All the species here with stars next to them are potential endophytes. Again, we don't know what their function is, but these are fungi that we can consistently isolate and grow in pure culture. The mycorrhizal fungi, shown here in italics here, Hebeloma, tuber, etc., a lot of these are either very difficult or cannot be readily cultured in pure culture. And so we've focused a lot of our initial studies on the endophytes. As you can see in Quercus and Pinus, however, virtually all of the top fungi that we detect using next-gen sequencing are ectomycorrhizal fungi. And I'll show you later on the pine ecosystems in particular are heavily dominated by ectomycorrhizal fungi. So this is sort of the most recent result of our bioassay work, but nowadays using Illumina MiSeq, amplicon sequencing protocols, we can basically look at hundreds of replicated community samples grown in a common greenhouse environment, and we can characterize, in this particular experiment, we looked at the 4 different common garden sites along with one of our North Carolina sites. And as you can see here, the primary pattern here is one of geographic provenance. So the soils from a given region are more likely to share similar fungal communities associated with, in this case, a single plant group. When we use different genotypes of Populus, there's no conspicuous pattern, although there might still be some heritable elements here. But you can see here also that there's no clear pattern of host association, except that the pines tend to cluster in one part of the distribution here, and Populus is in another region. So once again, we see differences between pine and Populus here. So as I mentioned, endophytes account for the majority of fungi that are associated with Populus. Some of the common groups that we find are Hypocriella and Ascomycetes. A lot of these are things like Fusarium. These are potentially pathogenic fungi that are growing within the plant root. Again, asymptomatically, maybe they're laying low or something, but they are consistently present within these otherwise naked Populus roots. We've also been detecting lots of this novel group here known as the Atractiellales. The Atractiellas are poorly known. There's just a few isolates known. In fact, most of the isolates that are known now are from Populus. And this appears to be a very common endophytic fungus within the Populus rhizosphere in particular. And then there's a host of ectomycorrhizal fungi present as well. But again, as I mentioned, we don't see them as frequently. So one of our lab's contributions to the project has been to isolate these fungi that we're able to. And we've got a collection of about 1,800 isolates now. Most are endophytes, and they represent only about 15% of the species that we can detect using next-gen sequencing. We've used these isolates sort of as a guide to select which ones should have their genomes sequenced by JGI. And in conjunction with a number of different CSPs, we've sort of been working our way through this fungal phylogeny of our culture collection to basically contribute isolates for genome sequencing so that we can proceed to do metagenomic studies with these fungi. Now, I mentioned the endophytes. We don't really know what they're doing. In fact, we don't even know if they're endophytes. So one priority has been let's try to re-inoculate these fungi onto the plant roots and then re-isolate them so that we can confirm that they are in fact growing within the plant roots. So we're using a sort of variation of Koch's postulates here to take pure cultures, grow up spawn, and then use that spawn to inoculate the soil in which we plant these plants and see what the effects are. And one of the things that we've observed very commonly is that many of these fungal endophytes actually have the ability to stimulate plant growth and plant fitness, although we don't know exactly how they do it. And so this is one of our results. Here's a variety of different oak seedlings in this particular case, inoculated with different strains of Mortierella. And as you can see, they do grow faster when these fungi are present in the soil. We know that these fungi do get into the plant roots as well. We also see higher rates of photosynthesis by these sort of beneficial endophytic rhizofungi. This is just another example. So Mortierella, one of the common soil fungi that we've isolated from plant roots, not only stimulates growth in Populus, but also in a variety of other plant systems. And so here's some just greenhouse studies using corn and tomato. You can see that Mortierella definitely makes plants grow faster and better. But not every endophyte does that. And some of these hypochrealian Ascomycete endophytes in particular show their true selves. When you inoculate them back onto their host, they actually decrease the growth of the host. So there's definitely some plant pathology involved in these endophytic interactions as well. And if anybody here is working on Arabidopsis, and I know there are, we've also put some of these strains onto Arabidopsis roots. And Mortierella, you can see here that the fungus actually migrates to the roots of the seedlings here and aggregates around those roots and stimulates growth. And in the case of Arabidopsis, not only stimulates growth, but also increases the plant's ability to respond to stress. So as I mentioned, we've been taking our fungal culture collection now, prioritizing these fungi for genome sequencing. And to date, we've completely sequenced 3 genomes, and we have 9 others that are on the way that have been submitted through various programs, the 1,000 fungi and so forth, fungal genome projects to have their genomes sequenced by JGI. I'm just going to mention one as an example, Mortierella. This was the first genome that we submitted for sequencing. This is one of the basal lineages that Mary mentioned, the Zygo1 group. And it's probably one of the most easily cultured fungi on the planet. I mean, if you culture soil, you'll get Mortierella, I guarantee it. It's an important fungus. It produces copious lipids in culture, and these are a valuable source of polyunsaturated fatty acids for baby formula, for example. And we also got a bonus here. This fungus harbors a number of different bacterial endosymbionts. And since we had the genome, we went ahead and we are doing some characterization of these endobacteria as well. Just wanted to show here, this is a corn plant grown in a clear gellan. And you can see, after this experiment was completed, Khalid Hamid from my lab, he just injected some trypan blue into the gel to see what would happen. Trypan blue is used as a fungal cell stain, and after about 2 days, all of that stain becomes localized around the roots of the plant. And if you zoom in, you can actually see that there's just a mantle, sheath of fungal mycelium that zeroes right in on these plant roots. So even though Mortierella is generally considered to be a saprobic soil fungus of no consequence, it appears to favor the rhizosphere, and we know that we've isolated it from within the plant roots themselves. We've done a variety of metabolic comparisons with it, and using biolog plates, we can very rapidly assess the types of nutrient requirements that it has. And interestingly, for example, it's able to utilize acetylglucosamine, and there's evidence that it is, in fact, a chitinolytic fungus. It does not utilize cellobiose. In fact, it's not a cellulose degrader at all. So it's quite unusual. But, and then also interesting, it uses tween as a nutrient. So it's able to utilize fatty acid analogs as a nutrient source. And in fact, we're starting to think that Mortierella is all about lipid metabolism. So a little bit more about the endosymbiont, just briefly here. The industrial mycologists reported recently that there are endosymbionts in Mortierella. And these endosymbionts are known to affect the lipid production of lipids by these fungi. So we characterized our endosymbiont, and we found that 16S sequencing puts it as a sister group to the genus Burkholderia, a very common plant-associated, fungal-associated bacterial group. And when we look at the genome of this endosymbiont, we've been unable to culture it. And in fact, we know that the genome of the endosymbionts are reduced. And so we see here, these are obligately biotrophic endosymbionts of Mortierella, and they are most closely related to another endosymbiont that exists that's known for the arbuscular mycorrhizal fungi, Rhizophagus, or the Glomeromycota. And thus the name Glomerobacter represents this particular lineage with a reduced genome and reduced metabolic capability. So it's effectively an endoparasite of these fungi. And we have discovered now that it seems to be widespread among these early diverging lineages of fungi. Not universally present, but certainly among those basal lineages. Interestingly, a number of metabolic pathways in the endosymbiont have been completely erased, such as the pentose phosphate shunt. And even glycolysis itself, basic glycolysis is impaired as well. So these endosymbionts clearly rely on their host for their nutrition. And the strongest evidence of that occurred when we cured the fungi by using antibiotics and then looked at their behavior. And this is work by Tim Chaplinsky at Oak Ridge, looking at metabolic profiling of these fungi with and without their endobacteria. And you can see here on two different media, the list of metabolites. These are almost all lipid metabolites. So it's almost, we're virtually certain that this particular endosymbiont is feeding and affecting the lipid production of its host. In fact, this was the basis for a recent CSP proposal that we submitted and that was funded last year to look at these early diverging endosymbionts within these basal lineages of fungi. And we'll have more to say about that soon, I hope. So the rest of my time, I would like to talk about this other project that we've worked on. As I mentioned, Populus, Trichocarpa, and Deltoides, at least, are not very strongly ectomycorrhizal. But pines certainly are. And the Pinaceae is one of the dominant forest biomes of North America. And so together with Tom Bruns and Kabir P. and John Taylor, we recently obtained funding from NSF, Dimensions of Biodiversity program, to look at the fungal communities associated with pine forests across North America. And you can see there's a Google map showing the sites that we've been able to sample from thus far. And the sampling scheme here is one that's designed to address the spatial elements of diversity. So the basic question is, do the numbers of species, how do they increase as you increase the scale of your sampling? And how does the scale of, how does function change with that scaling as well? And so these are just some recent results that Kabir's lab has published with Jenny Talbot as lead author. And you can see here there's clearly a pattern of geographic segregation that I also showed with the Populus communities as well. So I think it's quite clear now that we have discussions in microbiology about biogeography and dispersal limitation and so forth. It looks like fungi clearly have dispersal limitation. So fungi from the eastern part of North America and those communities are distinct from communities on the west coast and also distinct from communities in Alaska, for example. By contrast, the functional profiles of these communities, in this case based on soil enzyme profiling, really don't change, are the same everywhere. And in fact, water availability and other environmental factors have much more to do with those enzyme profiles. Okay, the last story I want to really just tell you about here is that we've been able to use the pine fungal symbiosis here with Ectomycorrhizae to look at host specificity involving specific groups of mushrooms that are known to be only associated with different pine species. And the genus Suillus, if you're a mushroom collector, I know that this is one of your favorite mushrooms because it's everywhere. If you're in the pine forest, you always find Suillus species fruiting. And several of these species, notably these three down here, Suillus granulatus, pictus, and americanus, these three species are only found under white pines. So whenever I see these fungi fruiting, I look up and I look where's the white pine growing, because it's invariably present. And so we mined our next-gen sequence data set to see where the suilli were occurring from the study I just described, and we can describe their species ranges using the next-gen data. And when we cluster them, we can see that in fact the white pine specialists specialize with white pine. There's a group of species that specialize on loblolly pine. And then there's other fungi here, other Suillus species that appear to be generalists. So we have a range of host specificity for the fungi on different pines. And with Suillus, we have the added advantage that we can culture this particular ectomycorrhizal fungus. And we can germinate spores. So we can take basidiospores, inoculate them onto pine seedlings, grow them for a short period of time, and then study their mycorrhizal interactions. And we've used this system to study their metatranscriptome and identify fungal genes associated with ectomycorrhizal symbiosis. So typically what we do then is we collect either pine roots or soil, isolate RNA and DNA, and then we characterize the transcriptome using Illumina RNA-seq. Now, when we do these experiments, you can have two possible results. You can get compatible formation of mycorrhizae, and this is what healthy mycorrhizal root tips look like with Suillus. And here's what incompatible pairings look like. Not much different than a naked control root. Interestingly, even these incompatible pairings still have fungal mycelium growing around the root, but you don't have a compatible mycorrhizal formation resulting. So here's the table summarizing the results for those white pine associates. You can see that the three white pine associates, they love to associate with white pines, but they cannot form mycorrhizae at all with loblolly pine, for example. By contrast, Suillus decipiens loves all the hard pines, but it really doesn't like the white pines. So we see evidence, in this case, just based on ectomycorrhiza formation in the flower pod, in the containers. So my postdoc, Sonny Leo, developed a pipeline for basically analyzing the metatranscriptomes of these mycorrhizal root systems. And we can do a number of things with this informatic pipeline. We can, first of all, we can go directly and start mapping and identifying the fungi that are present based simply on their ribosomal genes that leak through the high-seq run. Okay. We can also then do direct mapping to known fungal genes to identify what they're doing. And in the case where we don't have the ability to map them, we can also synthesize de novo reads, de novo assemblies, and then proceed to put together these transcriptomes. And we can do the same thing for the plants. So now we can simultaneously examine fungal and plant gene expression at the same time. And when we look at the compatible versus incompatible interactions, here we have simply the abundance of fungal transcripts shown in orange. You can see in the compatible pairings, almost 50% of the transcriptome from the root, from these mycorrhizal root clusters, are fungal genes that we can map to the Suillus genome. And the white portion shows the plant genes. We can map those, including the plant genes in the control. So we can also have uninoculated controls. You can see that the incompatible pairings do express a small level of fungal transcripts, but they are largely incompatible. So they're not forming a symbiosis. So I'm just going to show you 3 more slides here depicting what kinds of patterns we observe. The first type is what happens, how do we know that we're getting symbiotic interactions? So we can compare the compatible roots with the uninoculated and control roots, and we can also compare the fungal cultures of the fungus grown in isolation in order to see what those transcriptomic profiles are like. And so if we look at it from the fungal standpoint, you can see that ectomycorrhizal roots have a very different trajectory in terms of their transcription profile than fungal cultures, shown here. And so that's nice because there's somewhere on the order of about 300 fungal genes that are heavily upregulated in response to ectomycorrhiza formation. What about the plant? What is happening here? Well, a naked plant root has one profile, and ectomycorrhizal plant root has a different profile as well. So there, again, major transformation in the array of genes that are turned on during ectomycorrhiza formation. What happens when we have different Suillus species inoculated onto different pine species? Here again, we find sets of genes that are unique to each fungal species, and that's shown on the left here. So here's the same fungus, Suillus americanus, on 2 different pines, 2 different white pines, and here again, there's a different set of fungal transcripts that are upregulated depending on which host they are presented with. And if you look at it from the plant's perspective, it's even more interesting. This we didn't expect at all here, because here we see that you put different fungi onto the same host, and you see that the plant can actually recognize each fungal species. So if you're not a very good fungal taxonomist, you can let the plant tell you which species is which. Interestingly, and I'll show you in just a second here, virtually all of these upregulated fungal genes are various types of effectors or transcription-regulated genes. And so we know that there's definitely a developmental switch here that's happening in the fungi. What's happening in the pines? We don't know yet. A number of these genes are quite novel. The pine genome has not been completely assembled yet, but we can start mapping those. Another contrast, very interesting contrast, is what happens when you have compatible versus incompatible pairs of mycorrhizal symbionts. And as you can see here, there's a whole array of genes that are turned on in the incompatible response in both the fungus and in pines. And in this case, all of those transcripts map to known genes of known function that are associated with very particular responses. In the case of fungal incompatibility, what the fungus is doing here is it's secreting proteins and other effector molecules that are trying to induce a symbiosis, but they're not succeeding. In the case of the plant, when the plant incompatibility response reflects genes involved in plant immunity. And so I think that Francis Martin's group has also shown this in Leucaria. The MISP7 effector, for example, secreted protein, is involved in basically overcoming and turning off the plant defense response in compatible pairings. I'm going to just get to the summary here. We've looked at a number of these Sewillis, pine transcriptomes now, and we're reaching the point where we can identify a core set of genes that are involved in symbiosis, the ectomycorrhizal symbiosis involving Sewillis. We can also identify a core set of genes that are involved in the incompatible plant defense response that results in no mycorrhizae being formed. And when we look at these, if you're a plant molecular biologist, you'll certainly recognize some of these genes. But for the fungi, virtually all of these core genes are involved in regulating fungal transcription. These are regulatory proteins, G protein coupled receptors, and so forth. So we're just beginning to kind of have a look at these responses, but the general patterns are not that different from the ones we see involved in, for example, plant pathogen interactions. And so for example, in a compatible ectomycorrhizal interaction, you have a variety of plant ligands that induce a G protein signaling cascade, including a lot of these proteins have all of the signal peptides for transport to the nucleus and so forth. And likewise for the incompatible interactions. So I think I've gone way too long. Thank you very much.
All right, thanks. That was a great talk. Do we maybe have one quick question before? I have a question just about the biogeography observations. You saw this pattern that the fungi were so specific to the region. Do you still see that in the common gardens? And whether you do or not, does that mean that maybe the way those common gardens are started might have a big impact? Whether you're planting seeds or grafts or.
So the results I showed were using soils from the common gardens. And so those soils, have high fidelity, in terms of.
And are the trees started in that soil, or are they started in another soil?
No, the trees are started in that soil. So we're using cuttings. We surface sterilize the cuttings, and we put them directly into that soil.
So environment,
for sure.
Okay.